All right. Can you guys hear me? Yes, I guess. So let me first thank the organizers, member from the board. Has been a terrific meeting so far, and I'm looking forward to meet many other of you. So this is a painting from Peter Rubens, which actually was one of the most prominent artists, Flemish artists in the Baroque area. And this is Prometheus, a Titan, which essentially stole the fire from the gods, from the Olympian gods, and gave it to humankind as a way of knowledge, as a way of science, and as a way of technology. A champion of humankind, and perhaps people claim that he's the father of arts and human arts and science. Now, Zeus was not very happy with that, and what he decided to do is he deterred Father East, and Prometheus was tied into a rock, and during the day there was an eagle who will come and he will eat his liver. But during the night, the liver will regenerate. And this is a process that took forever, and in my view, this is sort of perhaps one of the first incredible examples of resilience, which is actually that ability to withstand in response to stress and challenging conditions and adapt. And when I talk about resilience, eventually I will be talking about pathways that regulate homeostasis. So in my lab, we are interested in understanding the basic mechanisms underlying resilience. And the way that we want to understand it is because perhaps we can mine it, and we can develop or reverse disease in a discovery-oriented but disease-agnostic manner. And as you will see, we are not targeting a disease. We are trying to understand the biological process underlying resilience, and perhaps, as you will see, develop new therapies. So I have to admit that I have a very holistic version about health, and I think the hallmarks of health essentially are characterized by the organization of the systems. And we have several system levels. So as you know, we are not only human cells, but we have actually more microbial cells than human cells. Those microbes, say, in the gut, could produce specific molecules that can circulate into the blood and change how a particular molecule is metabolized in the liver, or, as you will see in the second part of my talk, molecules that will go to the brain and affect essentially how the brain works. We have organ systems that have been discussed. Primarily, we talk about the brain, but in the brain we have different cell types, like neurons, astrocytes, glia, oligodendrocytes. Inside of those cell types, we have subcellular organisms. Here there is a mitochondria, which is an essential subcellular molecule which is required to produce energy. And of course, as we hear, we have micromolecular systems. We have RNA and proteins. Inside the cell, we have specific processes which are required to control the health of the cell. We have essentially a machinery that regulates how proteins are regulated. We have metabolic regulation, gene expression which is required to change the function, epigenetic changes that will allow to keep a memory of the cell, and a mechanism that's going to prevent us to accumulate DNA damage. And this is I will focus on proteostasis network, but as you could see, each of those processes are intertwined. And if you were to define disease, at least in my world, will be a progressive failure in the homeostasis and resilience of the system. And what I'm describing here is a very linear approach, but essentially each of those nodules, they have different negative feedback loops that are intertwined in a very complex manner. And what I will tell you today, I will tell you that I can touch each of the nodules either in the top or on the bottom, and by doing so, I can change perhaps the cell of the organism, increasing or enhancing abilities. So at the micromolecular system, I will focus on memory formation. As we have discussed, we have from Rodrigo, is essential for animal survival, is at the center of our identity. Here you have a description of a variety of cognitive disorders that strike the brain, say during development, like in the case of Down syndrome, or during adulthood, like in the case of Alzheimer's disease. Despite the high incidence that we have, effective treatments actually remain very limited. What we have failed as a field is to try to identify a common root for all those mechanisms. And I would like to leave you with this question: what will be the impact in human society of a successful universal treatment for memory loss? The reason why I was telling you about this is because Rodrigo mentioned that the consolidation of memory takes place in a particular brain structure, which is called the hippocampus. We have two different types of memories, at least. Memories that last just a few minutes, hours, and others last for days, months, and even a lifetime. What molecularly differentiates these two processes, what molecularly bypasses a long-term memory, is the requirement from protein synthesis. And the reason why I'm telling you about this is because when I was a postdoctoral fellow in the laboratory of Nahum Sonnenberg at McGill University, we discovered this factor, which is a memory repressor molecule that determines whether a short-term memory is converted into a long-term one. If we mutate the phosphorylation of this factor, we can form memories or long-term memories in a much easier fashion, and I will be happy to tell you how we do that. And the opposite is also true. If we were to increase the phosphorylation of this factor, now we can form long-term memories. And this has been expanded by many investigators around the world. Most remarkably, my colleague, now at Altos, but before at UCSF, discovered a molecule which is called ISRIV, which essentially does the same thing that the genetic inhibition of this factor that is able to enhance memory. This has affected the learning and memory field, but also industry efforts. And here you have some of the major pharma which are targeting this pathway, which is called the integrated stress response. I'm not going to go into details what this pathway does, but this is a pathway that senses changes in the environment, and it has dedicated sensors which converge all the information in this factor. And when this factor is phosphorylated, the synthesis of proteins actually is inhibited. For instance, when you have proteins which are misfolded, it doesn't make sense for the cell to synthesize more proteins. So there's an inhibition of the synthesis of protein, but at the same time, paradoxically, there's an increase in the synthesis of proteins that will help to resolve the problem and will help to fold those proteins. If the cell cannot cope with that stress, actually the cell will die. And I have no time to go through all the details, but for all the diseases that we have in there, we have a causal evidence that essentially in all those diseases, there's an activation of this pathway, an increase in phosphorylation of this factor, and now this leads to a memory impairment, but perhaps we have ways that we can use to reverse it. So why should we target this pathway? We should target it because it is a central memory switch. It's conserved through evolution. It promotes health and function by resetting the homeostasis of the cell. This regulation is associated with cognitive decline, cognitive and age-related diseases. So why to do it now? Because the findings that we have are orthogonal, and the evidence is super strong. It doesn't matter at which level of the pathway that I inhibit the ISR, invariably, or using genetics or pharmacology, or if I do it in a single cell level, invariably when I inhibit the ISR, I am able to essentially enhance cognitive abilities in animal models and in chicks as well. So how can we optimize success, or our chances of success? Okay, so I will use an example of a game which is played mostly in Europe and in South America. And the goal is the objective is of that ball to essentially get into that goal. And what many of you are doing, as we have heard from Francis, is increasing precision. You can increase precision and you will increase your possibility of success. As you will see, we have a different way of thinking, and we want to put more balls, but also we want to put more goals and see what that will result into it. So the phosphorylation of this factor indicates that the animal is unable to form a memory, and there is a molecule which removes this phosphorylation site, and there is a scaffold protein that links these two processes. And in here, what you have is you have two cellular proteins that contains those two domains, the binding to PP1 and the binding to EIF2. So what we did is, as you may know, the biology space is characterized by innovations, disruptive innovations, which result from evolution. So what we did is we went through evolution and we wonder, is it molecules in, say, viruses, which contain these two domains? And we found a viral protein, which is super small, which contain these two domains. And then when we now inject it into cells, and this pathway is activated, just compare the second lane with the 4 lane, the second lane with the 4 lane. Regardless of the kinase that gets activated, invariably, this viral protein is able to inhibit the integrated stress response of the phosphorylation. The beauty of it, as Francis mentioned, AAV is a way, or perhaps the best way that we have in the field for gene therapy. But what he didn't mention is that we are limited by the amount of genetic information that we can include into it. But if you were to have a viral protein which is super small, you can put it in AAV, you can infect the brain, and now you can test whether this could be used as a way to reverse memory formation. We use a particular task, which is called fear conditioning, where the animals will associate a particular environment, say green, with a food shock. And when you put it 24 hours later and we measure the long-term memory, they will freeze because we'll associate that environment, green, with an aversive experience. What you see here on top is animals, Down syndrome animals. Those are animals that they don't have an extra chromosome 21, but they have a chromosome 16 and 17 translocation, which contain about 80% of the genes which are duplicated in chromosome 21. And what you could see here, those animals, they freeze less as an indication that their memory is impaired. But if we were to inject this protein in the brain, we are able to reverse the cognitive decline. If we were to measure the strength of synaptic connections in field recordings, no health recording, those are long-lasting changes in synaptic function. We can see that in the mutant animals, those changes are deficient. But if we inject this protein, we are able to reverse it. At the bottom, you have animals that they have Alzheimer's disease, or they have a mutation that in human has been associated with Alzheimer's disease. And like the patients, the human patients, cognition is actually impaired. But now if you were to silence the ISR, you are actually able to reverse it. So this is one ball. By the way, if you are interested, I can tell you that we are also able to enhance memory and plasticity in normal animals, but we can discuss with Rodrigo actually the benefits and advantages and disadvantages of doing this in normal individuals. We are incredibly excited about this because there is 440 other proteins in the animal kingdom that have those two features that we can mine and essentially perhaps take those secrets from nature. Nature has done all the experiments, according to Francis Arnold, that we can use it for therapeutics. And of course, we have also, in the case of this protein, an anatomical understanding of how this protein is functioning. And there is this protein compared to the cellular protein has a new amino acid change, which is predicted to make a new hydrogen bond. And actually, and that's the reason why the protein is so effective, even much more so than a cellular protein. But what about a second ball? What about molecules? What about drugs? And this is my colleague, Peter Walter, who came with an incredible molecule. When this factor is phosphorylated, it will inhibit this pentameric subunit. So this decameric subunit, which are 2 pentamers. And as you could see here, you have 2 yellow subunits, 2 red subunits, 2 blue, and so on. And the molecule, the protein is perfectly symmetrical. In the middle of that protein, you will see a pocket. And in that pocket, there is the molecule that Peter discovered, which is also perfectly symmetrical, as you could see. And when the molecule binds, it makes a small movement, opens its wings like a butterfly. And by making this movement, it's able to remove this factor which is called EIF2. And that's how essentially it's able to reverse the effects of activation of the ISR. And here we have some of our data that we have used this compound, again in Down syndrome animals, and we are able to reverse the cognitive decline in those animals. Now, so what I told you now in the first part of my talk is that perhaps there is a new concept that could explain cognitive disorders. And the concept is that you have a disruption in proteostasis. If you think about it, if you have a deletion in chromosome 2, or say insertion in chromosome 7, you have cognitive decline. If you have Down syndrome, an extra chromosome 21, you also have cognitive decline. The genes that they are in chromosome 2, 7, and 21, they are very different. So it cannot be genetic. However, what they have all in common, that this leads to a change in the proteostasis of the cell. When that happens, this pathway, the ISR gets activated. And when the ISR is activated, you can no longer form long-term memories. Well, actually, you could if we were to develop or push some of the technologies that we are creating to essentially alleviate the cognitive decline associated to those diseases. But now I will tell you that perhaps we can promote health or enhance at least animal abilities, even human abilities, as you will see, by essentially looking at, by modulating microbes. And as you know, we have more human microbes that start in the Milky Way. They largely outnumber human cells. So if we were to count the number of cells that we have, we are more microbial than human. And every single time that you go to the toilet for number 2, you became more humanized. And you should think about it every single time you do it. Microbial genes outnumber our human genes, and those microbes, for the most part, are actually beneficial for us. Now, where those microbes emerge, or what is the relationship with those microbes have with the nervous system? So this is Hydra. Belongs to the phylum Cnidaria, was generated about 500 million years ago. Is the first organism that has a nervous system, and the behavior that this animal has is a sort of a contractibility which is required for morphogenesis, regeneration, osmoregulation, and respiration and propulsion. So this is not a very complex behavior, but nonetheless, this is what this animal has. And which is remarkable, if you were to eliminate the genes, the microbes in this animal, you are essentially able to change the behavior. And to me, this is the first example in evolution in which microbes are able to change behavior and actually talk to the nervous system. The reason why I'm talking about this is because what I will show you is the result of a serendipity discovery in my lab. And a postdoctor in my lab, Shelley Buffington, was interested in autism spectrum disorder. And autism spectrum disorder is characterized by a lack or a deficiency in social behaviors, repetitive behaviors, and language problems. And we develop a model, and we measure behavior in the way in which a mouse has to decide what to do, either spend time interacting with an empty cage or spending time interacting with a cage in which there's another animal. And if you were to be a mouse which is normal, as you could see here, the control mice, they spend most of the time interacting with the mouse. However, models for autism, they are socially impaired. But this is where serendipity struck. If we were to combine three of the control animals with one of the disease animal, the behavior is actually corrected. And it doesn't matter the ratio, 3:1, 2:2, 1:3. Every single time that you put together a diseased mouse with a normal mouse, the diseased mouse is cured. And we say, what the heck is going on in here? How can that be explained? And to make a long story short, the reason that this is explained is because the diseased mouse lacks a microbe which is required for the social behavior of the animal. The microbe that lacks is called Lactobacillus reuteri, We went, we sequenced, we tried to identify it, we did identify it, and when we put the microbe alone, we are able to reverse the social deficit in this animal. It's one microbe, but not any microbe. If we take a cousin of L. reuteri, L. johnsoni failed to reverse the social deficits. Now, I have to say that in my lab and in many other labs since this original discovery, we have found that using this microbe, we can reverse social deficit, only social deficit, or the social deficit in multiple preclinical models of ASD. And it could be genetic, it could be environmental, or it could be idiopathic. And I'm sorry, I'm strictly speaking about mouse models of autism spectrum disorder. Some of you may ask mechanistically how a microbe in the gut is able to modulate the brain. And the answer is the following. The microbe is able to induce a hormone which is called oxytocin, the so-called social hormone, in a particular brain area which is called the paraventricular nuclei. The paraventricular nuclei has connections with the area on the brain that Jose told you before, which is called the ventral tegmental area. There's an area that senses reward. In this particular case, it's sensing social reward. What we found, if we put two animals to socialize and we put an electrode in those neurons will fire in a similar way if you were to give to the animals cocaine, amphetamine, or any other drug of abuse. However, in those animals in which they have, say, autism spectrum disorder, this bacteria is eliminated from the system, and as a result, the entire circuit is actually turned off. And if you were to put those animals to socialize, that area of the brain doesn't light up. And we believe that this is the major reason why, essentially, kids, they are not interested in social interactions. Now, I have to say that the signal goes through the vagus nerve, and we have found essentially a molecule that is induced by the bacteria that if we were just to give the molecule alone, we are also able to reverse the social deficit. So now we have some good news for you, and especially if you are a mouse and you have autism, we may have a way to do this, to reverse your social deficit. But, you know, science is remarkable, and during the pandemic, there is a group in Italy led by Luigi Mazzone, which actually they have done a clinical trial in kids with autism. And he called me right before Christmas, and he told me that he wanted to share with me the results. I didn't believe him, so I contacted Antonio Hardan, which is an expert in clinical trials from Stanford, and I asked him, would you mind to analyze the data? And the data invariably shows what essentially we show in mice, that is, that if you were to give the bacteria to kids, it's about 20 kids, double-blind placebo, we are able to reverse the social deficits in those kids. And there is another paper that is in the bioarchive, which are much, the numbers are actually not so big, and actually the conclusion is the same. And from my recollection, this is the first example that essentially structures outside of the brain that are able to modulate brain function in a powerful manner, not only in animals, but now in humans. And specifically, I am talking about the microbiome. So where can we go from now? And this is my last slide. We hear from Jim Collins, could we develop synthetic bacteria, next generation of probiotics? We believe that we know what is the gene, the social gene in this bacteria, which is driving the behavior. Can we put 4, 5, 10 copies and have a super social bacteria? Can we identify the metabolite that this bacteria is producing and give it to people? The advantage of any of these technologies that eventually is non-invasive at all, as you can imagine. Can we develop synthetic communities that can promote brain health? Or can we develop foods which essentially make the good bacteria grow and kill the bad bacteria that we have in the gut? This is all essentially in the future. I want to leave you with my last slide. This is how my lab operates. We have absolutely no idea what we are shooting into it. But the beauty is if you shoot first, you are the one who drives the target. And I will be finished by thanking an incredible group of people that I have I'm happy to answer any questions that you guys might have. Thank you very much.